Guys, welcome back to my YouTube channel. Today we are going to discuss another very important topic from immunology. As you can see, we'll discuss about antibody and its structure. What is antibody? It is a structure that binds with antigen in order to disarm it. Disarm means destroy it or to decrease its effect over the body. Okay? So, sorry. It is made up of glycoprotein. Its structure has two heavy chains and two light chains. Okay? You can see, you might have seen this structure earlier many a times. Now let's discuss the structure in detail. The antibody structure has one N terminal and a C terminal. Both the chains, heavy chain and the light chains, have both the terminals, that is, one is N terminal and the other is C terminal. N terminal has a amino group as its functional group, and C terminal has a carboxylic group as its functional group. The N terminal has the antigen binding site. Okay. Both heavy and the light chains are attached with each other by a covalent bond, that is the disulfide bond. You can see here it is attached by disulfide linkages. Okay, there are 16 disulfide bonds. You can see that there are 16 disulfide bonds. Now let's mark them all. Which all? 16. Here it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 and 16. Please remember these. This is very important that each antibody has 16 disulfide bond. You need to learn out the locations. Okay. Antigen binding site in the variable region is known as paratop. I told you this is the antigen binding site. This is known as paratop. Okay. And the site to which it binds is on the antigen surface is called as the epitope. Okay. Okay. Hinge region leads to the flexibility in antibody structure. This is the hinge region. You can see it is not straight, it is somewhat wavy. This is the hinge region, and it is such because to provide flexibility in an antibody structure. It is generally of 10 to 60 amino acid, rich in cysteine and proline. Remember, these are very important, that hinge region is rich in cysteine and proline. Okay. Now, basically, all the light chains and heavy chains are divided into two regions majorly. One is the variable region and the other is the constant region. Variable region, why it is termed so? Because it varies from antibody to antibody. Basically, it shows that different antibodies bind with different antigens. Okay, so this region varies with antibodies. So the variable region is marked as VH because it is the variable region of heavy chain, and variable region here is marked as VL because it is the variable region of light chain. Here again VH because it is of heavy chain, here it is VL because it is of light chain. Okay, then the rest below that comes the constant region. This region is constant, and this determines that which type of antibody is shown by the structure. Okay, so here we have marked it like CH1, CH2, CH3 and Cl for the constant region of light chain. Okay. The structure is quite easy. You will have to practice 2 to 3 times, then you will get up to it. Okay. So the region, the above region is also referred to as FAB. That is fragment of antigen binding. Okay. Remember, it is called the fragment of antigen binding, and the below wall structure is called the FC region. That is the fragment of crystallization. This was demonstrated experimentally. You just need to remember that the above structure is known as the FAB region and the below structure is FC region. Okay, weight of heavy chain is 50 kilodalton, light chain is 25 kilodalton. So overall, if we consider, then it is nearly 150 kilodalton of an antibody structure. Okay, and if we consider the amount of amino acid, it is nearly 660 amino acids, right? Okay. In a light chain, the variable and the constant region both are made up of 110 amino acids. I've told you earlier. In a heavy chain, variable region is of 110 amino acid and the constant region is 330 amino acids. Variable region consists of a hypervariable region. This is very important. Variable region consists of a hypervariable site which changes according to antigen. Okay, the antigen also sometimes determine. I told you the antibody's variable region changes according to the antigen it is going to bind with. Okay, so the hypervariable region is generally determined by the antigen only. Hypervariable region consists of CDR. CDR consists of 110 amino acids. CDR stands for complementary determining region. Okay, it divided into CDR1, CDR2, CDR3. CDR3 is the most variable one. I told you FAB stands for fragment of antigen binding and FC stands for fragment of crystallization. Now we will discuss the antigen and antibody interaction in the next video. I hope you like the video and you don't. If you have any doubt, please drop down in the comment section. Thank you. Please like